Good morning or good afternoon, good evening, wherever you are. What I want to do in this talk today is kind of tell you a story about the deep ocean, its remarkable microbial life, some of the things I'm the most excited about, and why I think we need to study it. Given the diverse audience in this symposium and the fact that probably most of you aren't oceanographers, I'm going to keep this pretty high level, but hopefully interesting enough that you want to learn more. So let's dive in. I study microbial life in the deepest parts of the ocean, and this is one of my favorite maps of the planet, which is a little bit weird because the ocean has actually been subtracted out of it. But what I like about it is that you can actually see the seafloor and all the amazing features that cover 70% of our planet's surface. Most of the seafloor is covered in sediments, but beneath it all is the rocky ocean crust, the habitat that I study. And many of these features that you see on the seafloor are formed through the process of plate tectonics, where you have new oceanic crust being formed at mid-ocean ridges, or you also have plates spreading apart and getting hydrothermal vents, and you have plates spreading in, slamming into one another, and all sorts of cool processes happening that create volcanic activity. And in fact, about 75% of all that volcanic activity on Earth occurs underwater. And both at and beneath the seafloor, I study life, and in particular microbial life, associated with these crustal ocean environments. And you are all very familiar with this universal tree of life for all DNA-based life on our planet, showing the main domains. And the crustal ocean biosphere is really the diverse biosphere of both visible animals and invisible microscopic life hosted in crustal ocean habitats. And I mention these animals because in many deep sea habitats, microbes form symbiotic relationships with many of those animals. And microbes, of course, play a really important role in transforming carbon, nitrogen, other elements, and are thus a key component of ocean biogeochemistry. But studying life at and beneath the seafloor is really challenging. Less than 0.001% of the seafloor has actually been visually observed by humans, either through a robot's eyes or from a submersible. And that's about the size of the 10th of the size of the country of Belgium. And we've barely even mapped the seafloor at high resolution. In these habitats, less than 10% of deep sea animal species have been described, meaning they've actually been collected and taxonomically described. And for microbes, it's well less than 1%. So this is really a largely unknown part of our planet. So before we move on, I wanted to give you a better understanding of what the seafloor looks like with a movie. That will show you 6 very different places that I've had the privilege of visiting and working at on the seafloor. And hopefully this won't lag too much and I'll be able to speed right through it. But probably familiar to most of you are these spectacular black smokers, which are formed when seawater reacts with a heat source beneath the seafloor and is chemically modified. And then when all these minerals precipitate, when that hot buoyant fluid returns back to the deep ocean, producing these sulfide chimneys or black smokers like these in the Western Pacific near Guam. These hot fluids can also mix within the crust and produce much lower temperature fluids. Basically all this shimmery stuff you see in this video footage from Axial Seamount in the Northeast Pacific. And this is actually lower temperature and where you see most of the animal life on the seafloor, from limpets to that purple protestan map to, of course, these tube worms, which are thriving in this environment because they're chock full of symbiotic microbes that are harnessing energy from these volcanic fluids. This type of volcanism is probably less familiar. This amazing firework display is West Mata Volcano. It's located near Samoa in the Western Pacific. These 1200-degree lava bombs are creating this really acidic plume about pH 1 near battery acid. And when we first captured this video in 2009, no one had ever seen anything like this before. Also in the Western Pacific, but up in Japanese waters, this is the volcano Daikoku, located along the Mariana Arc. This is actually molten sulfur that is created when the subducting Pacific plate is remelting, where you get this otherworldly scene. And then finally, we have an example of a no longer volcanically active, but still really important rocky seafloor environment seamounts. This is a 20 million year old one in Costa Rica waters. And here you can see these mother octopus, which are brooding their eggs in these really, really low temperature fluids that are leaking out of the crust. That shimmer that you see is about 5 to 8 degrees Celsius fluids. And for reasons we don't totally understand, these mama octopus are brooding their eggs in these fluids for years, slowly dying until their eggs hatch and then they die. So hopefully those videos help you understand the diversity of rocky seafloor habitats on Earth. And again, many of these sites look different from one another because the underlying rock and tectonic setting is different. Thus, the nature of the chemical reactions are different. And that then changes what types of life can thrive. So everything we do in our lab kind of follows this mantra, which is kind of what is the basic process occurring between rocks, water, and life. You know, water and rock react. Water is flowing through rocks. It creates energy sources. And the question, of course, is can life extract that energy? I put the question mark because there are, of course, examples where there's plenty of energy, but some other requirements for life are not met. And those black smokers are a great example where there's chock full of energy, but it's 350 degrees Celsius and life obviously cannot live there. So specific questions that our lab here at Woods Hole Oceanographic addresses includes what are microbes doing here? What ecosystem services do they provide both to us and to the deep ocean? How do our own human activities impact those services? And how can the deep sea inform our understanding of the origin of life on Earth and beyond? And again, because of sort of the level of this talk and the diverse audience, I'm just going to touch a little bit on each of these topics. Now, everything I've been talking about so far is looking right at the seafloor, but of course, the ocean doesn't stop at the seafloor. The truth is, I'm also interested in what is happening beneath the seafloor, which is an even harder concept to convey since I can't show you what it looks like. I can't actually put a camera beneath the seafloor and show it to you. But this cartoon gives a really highly stylized version of what's happening beneath the seafloor. And you can kind of think about it as a giant aquifer with a large amount of water moving through this rocky ocean matrix, getting altered, and at some point finding a way out. And in fact, at about any single point in time, about 2% about the global volume of the ocean is trapped within the oceanic crust with a mean residence time of a few thousand years. So we have this really large habitable volume. We have water moving through it. We have nutrients. We have potential energy sources. We have constant water rock reactions taking place, which means we have this really large habitable volume for microbial life. And in the last 50 years or so, we've learned that there are microbes in the subsea floor across a huge range of habitats, and they play a really important role in regulating carbon flux, for example, and other important biogeochemical cycles. But we just know very little about the extent, the variation, and the impact of this varied community. And really, we just don't have that much data, especially from that hard rock environment and the fluids moving through it. So if we go back to this map that I showed you at the very beginning, and we kind of draw a line from the Philippines back to the west coast of the United States, and then we turn it on its side, we can take a peek at the different types of fluid regimes we might encounter in the deep sea. And you can see that as a result of plate tectonics, there's a lot of different sort of manifestations of fluids leaking out of the seafloor. And in fact, I've sampled almost all of these fluids in the ocean in a variety of places. They are chock full of microbial life. The simplest thing that I do, which is not in fact that simple, is I go to the seafloor and fluids that are naturally leaking out of the crust, I collect them and I count the cells under the microscope. And they range in concentration from a few hundred cells per milliliter up to 100,000 cells per milliliter of water. And in fact, it was from collecting water from vent fluids shortly after the discovery of deep sea hydrothermal vents in 1977 that even the idea of a subsea floor biosphere within the rocky crust was first hypothesized. Scientists had known for a long time, for example, that marine sediments had contained microbes, but no one knew that fluids were even leaking out of the crust until hydrothermal vents were discovered in the Galapagos. So there was really no concept of this rocky subsea floor until vents were discovered about 50 years ago now. But in the discovery paper, John Corliss and his authors talked about this flux of bacteria from the vents and how it must be supported by the productivity of a large population of bacteria living within the rock mass, lining the walls of the fissures through which hydrogen sulfide-laden fluids ascend. And both myself and many others have pursued this idea ever since the discovery of vents to really set the stage for the work to follow. And I think this initial concept is really captured beautifully by video footage from one of my favorite places on the seafloor, Axial Seamount in the Northeast Pacific. This is an underwater volcano that has erupted 3 times in the last 25 years. And this footage was taken shortly after an eruption in 2011. And what you're seeing is all this white snow from sulfide and sulfur oxidizing bacteria that are ejected from the crust during these large eruptions. And all this hydrogen sulfide and heat and energy is brought out, and microbes are getting flushed out of the seafloor. This is the collection device that we're using. And you can see microbes are actually just coating every surface. This is sort of like a reverse phytoplankton bloom, but it's at the bottom of the ocean, and instead of energy from the sun, microbes are using energy from the volcano. And we call these snowblower vents. These have now been seen a few times at mid-ocean ridge systems and is some of the best evidence we have of a subsea floor biosphere in young oceanic crust. So let's just kind of zoom in and take a look at this subsea floor and what it means for microbial life. Luckily, I'm assuming there's no geologists in the audience, but this is a terrible diagram if you're a geologist, but it's really important because as ocean crust ages, the parameters of how life has to get by change a lot. As you move away from the crust, from left to right, microbes have to deal with different things. The characteristics of the water rock reactions change. So the flow rates, the porosity or how much space is there, the energy available, the thermal gradients, the energy and the biomass and the carbon flux. And so shown here are how these broad characteristics might change as you move from a mid-ocean ridge off axis. So at deep sea hydrothermal vents, things happen very quickly. These fluids, as I showed you, gushing out of those smokers are very fast. And we also have a lot more measurements from these regions and just kind of know more, but they have high flow rates, because we have this heat source, we have big pressure changes. The crust is really porous, as that video showed with the snow blowers. There's a lot of space for microbes to move through it. And we have these really steep geochemical and thermal gradients. And that creates a lot of important redox couples for microbes to exploit. And you have temperatures from 400 to 400 degrees Celsius over a very small spatial range. And that means there's just a lot of energy for microbes to exploit, which means the biomass is quite high. These are really hot spots of primary production in the deep sea. And this is sort of all in contrast is when you get out into the ridge flanks, because by now the crust is sort of filling in, flow rates are slower. You do not have all this volcanic energy. Temperatures are much lower, and as a result of less energy, lower temperatures, et cetera, the biomass is lower, and instead of being a source of carbon, the few data points we have indicate this is actually a sink of carbon. And so while this isn't really the only thing I do in my lab, I think a lot about how microbes are distributed in these different environments, how active they are, sort of what's driving their evolution, and also both how viruses and grazers impact these communities. And we work at both a very, very small scale, sort of strain level genomic work, and also just big picture global, thinking about, how do these communities vary across ocean basins, across multiple sites, et cetera. And as I already mentioned, we're really data rich in the hydrothermal vent environment and relatively data poor in the ridge flank environment, although we have gotten a lot more data in the last 15 years from 2 sites in particular. We've learned a lot about how microbes operate in these vent ecosystems, and much of my career has spent in this bucket in the bottom of these microbial primary producers or microbial autotrophs, who are again fixing inorganic carbon and gaining energy from the chemicals in these venting fluids, and they really form the base of the food chain. And if you look at my Google Scholar page, a lot of my work is on developing tools and methods like RNA stable isotope probing and other things to try to really figure out what these microbes are doing, how they're doing it, and how much of it. And I want to just give you a specific example of what, a remarkable primary producer in these ecosystems look like. So this is a group of microbes that lives on Earth called a methanogen, and these are super common in deep sea hydrothermal vents. These belong to the domain Archaea, and especially predominant in hydrothermal vents are hydrogenotrophic methanogens that are taking hydrogen and CO2 and they're making methane. And these organisms can grow up to the highest temperature known for life on our planet, which is 122 degrees Celsius. This organism, an example of this organism, was isolated from a deep sea hydrothermal vent. So these are wildly successful organisms obviously across our planet, but this type in particular in the deep sea. But one of the big challenges, of course, of working in an environment, any natural environment, but particularly one where you have such a huge range of redox couples and unknowns, is how we study it. And so I was really lucky early in my career to sort of be on the cusp of when next generation sequencing was invented. And, we discovered sort of surprisingly that these environments had exceptionally high diversity when we looked at it phylogenetically. So this is work I did with Mitch Sogan many years ago, when we discovered this sort of long tail of biodiversity within the microbial communities throughout the world's oceans, but we first found it at these deep sea hydrothermal vents. The colors don't really matter, but what really shows is these remarkably high levels of microdiversity. And we believe, particularly in vents, and this is held up in the era of metagenomics, is just when you have such a wide array of energy sources and so much temporal and spatial change over short distance, you just have a lot of niche space to fill up, and things are constantly changing. But it isn't just that there are a lot of different taxa in this environment. They're remarkably functionally diverse as well. And this is what we've tackled with metagenomics, stable isotope probing, and also metatranscriptomics. They can just kind of exploit a huge range of growth conditions due to this mixing regime, from fully oxic and quite cold to microaerobic to fully anaerobic and very hot. And they can then couple these redox reactions to exploit that. And so this is an example on the left showing 3 different hydrothermal vents and sort of going from cooler temperatures down to higher temperatures. We often see these methanogens, for example, at higher temperatures. We see a lot of nitrate reduction, hydrogen oxidation, and sulfur oxidation. One thing we also see, though, is a lot of functional redundancy, or really different groups from different branches from the tree of life that are doing the same thing. And this is something we see in a lot of ecosystems, which is perhaps not surprising, right? Many lineages follow the energy, maybe not even with the same set of genes, maybe they are, but they're all sort of exploiting similar energy sources, but perhaps under slightly different ways. And another interesting thing that we've been finding is that while we see big patterns across different sites, this sort of plumbing and chemistry of the system is really impacting microbial distribution and activity. And I'm not going to show all this data, it's all been published, but we see both really large scale and small scale geographic patterns in microbial distribution, where quite often each vent, even within a vent field, is sort of like an island unto itself. And I've started thinking about these sites, even if it's just a meter wide, sort of like a burn hole, where sort of the heat of circulation and the vent system, it kind of gets trapped, and it's just sort of this vertical. There's not a lot of horizontal mixing going on. And this begs understanding a lot more about the hydrology and the physical structure, and the nature of the flow. And one thing that we've been working on with many others is sort of how do these microbes hold on? How do they get to these big effective population sizes that can maintain over time in that type of environment? And we think holding on through biofilms or other attachment is a really important phenotype that merits more study. And one thing in the last 5 years or so, once I kind of got a better handle on the bottom of the food chain, is that I then wanted to know, okay, so this is what's making all my carbon. Who's killing my primary producers, right, in this microbial food web if we start moving up the trophic cascade. And so I've been working with a group of really talented young folks who brought that skill set to my lab to look at both viruses and grazers, both things that eat or kill the microbes and do a lot of other interesting things as well. And one thing we've done is we've looked at eukaryotic grazing by protists. These are really hard experiments to do. You do them by labeling bacteria with a fluorescent dye and then feeding it back to the natural fluid and looking at the disappearance of your prey over time. We've done these experiments both on the ship as well as at in situ pressure to see if it's faster under natural conditions. The answer is yes, it's a little bit faster under high pressure, but it's a lot harder to do the experiment. And doing shipboard is probably just fine. And what this plot is showing is these are the rates of grazing over on the left. This is cells per mil per hour consumed. This is background seawater on the left. You can see the rates of grazing, and then all these measurements here are in these mixed fluids coming out of the seafloor. And so you can see these are really hot spots of grazing compared to the surrounding ocean, which makes sense given these are also hot spots of primary production. And as I mentioned, these in situ rates where we were able to do pressure, these are the upside down triangles. You can see these are some of the highest rate measurements we got, but we also got pretty high measurements even when we did them at 1 atmosphere. And so if you can't do them the fancy way, it's okay to do them the regular way too. You're still getting pretty good measurements that are statistically significantly higher than background seawater. Just like with our prokaryotes, we've been trying to understand the distribution of these microbial eukaryotes. As I mentioned, we see a lot of restricted distribution of prokaryotes in the deep sea by vent field, by vent site, by individual vent. And this is an example of a study by Sarah Hu, who's now a professor at Texas A&M. She was a postdoc in the lab at the time. And she basically looked at the 18S ribosomal RNA gene at 3 different vent sites in the Pacific Ocean as well as the Caribbean Sea. And basically, she found that these organisms exhibit endemism across these ocean basins, very similar to what we see in bacteria and archaea. And it's actually a little bit hard to see, but she found that, for example, there were some sequences that were only detected, for example, down here in this red bar, they were only found at axial. They were not found, at this site, which is off, only about, I don't know, this is off the coast of Northern California, whereas this is off the coast of Oregon. So we see that, that there's a lot of local endemic eukaryotic lineages at these sites. We also looked at the distance decay relationship, and we see that vents that are very far apart are very different, but even sites really close together have a wide range of dissimilarity. And again, it sort of highlights how localized at individual sites these patterns can be. And right now, Sarah and I have generated a ton of eukaryotic metatranscriptomic data from these sites, really trying to dig into the lifestyle of these organisms to figure out how are they maintaining this type of lifestyle in the vents, whether it's through parasitism, symbioses, heterotrophs, etc. We've also been looking at the other killer of microbes, which is viruses. And for viruses, most of the data is from DNA viruses. There's a little bit of RNA virus work that's just starting to come out from vents, but there still isn't that much of it. In this study, which was published a while ago, we looked at viral OTUs from hosts' metagenomes. And this was also from 2 sites, one in the Pacific and one in the Caribbean Sea. We know that microbes or viruses kill microbes. They're also really important agents of horizontal gene transfer. And we found very clearly that, these viruses they're incredibly diverse, and similar to what we saw in the previous slide with the eukaryotes, and also what we see with our prokaryotes, is that they're largely endemic to these individual vent sites. And again, this is just the DNA viruses collected in a metagenome. So this isn't a true virome, which is something where we and many others are working on as well. We also did an infection network to try to show the links between the viral clusters and their host. We created these hosts through metagenome, through MAGs, metagenome assembled genomes. And so the edges are the little dots where a viral OTU is then linked back to its host in the same sample. And what I hope you can just kind of see is that the viral host range is really narrow, with very few viruses infecting multiple microbial lineages. And so what we see is these viruses are highly localized to specific regions and specific taxa. And there are, of course, a few exceptions where we see a virus infecting multiple hosts, as up here, for example, although these are all within the same phyla or class, I'm not sure. But there just aren't a lot of generalist viruses out there in this vent ecosystem. But I told you already that the prokaryotic hosts themselves are pretty well isolated from one another. So it makes a lot of sense that we're seeing this in the bacteria and archaea, and then also in the things that are killing them. So a lot of specialization with some generalists as well. Now, at the beginning of the talk, I mentioned that I study ridge flank systems as well, but we have a lot less data. But I did want to mention it because a lot of the seafloor is not volcanically active. It is not a mid-ocean ridge. A lot of it is just older oceanic crust, and it's very important in terms of biogeochemistry and very important in terms of a large habitable volume for life on our planet. And so it's really important to study it. It's just a lot harder to do because it's covered in sediment, and you have to use specialized technology and ocean drilling, and that's a whole nother talk. I'll just mention that we have 3 sites, which are shown on the map above, where we have pretty good data. One is in Costa Rican waters. It's called the El Dorado system. One is off in Canadian waters called the Juan de Fuca system. And the other is in the middle of the Atlantic Ocean called the North Pond system. And in the table below, you can see that they range from 3.5 to 23 million years old. They have different fluid flow regimes. 2 of them are quite cool. One of them is a little bit warmer. And so they've presented a really nice opportunity to study microbial communities in a very different environment than a mid-ocean ridge. We've learned a lot from this study. From these studies, the main environment that my lab has been working in is this North Pond system, which is pretty young and cold, less than 10 degrees Celsius. And I've had a really talented group of postdocs, all who've now moved on from the lab. But we found basically this is a really low biomass community, about 1,000 cells per mil. But they're active. We can detect their mRNA. We can show that if we feed them a labeled substrate, they eat it. And we have done a bunch of time series work showing that they're scooting around within the aquifer and moving about. They're highly motile. We can see that they're eating both inorganic and organic carbon, but this is mostly a heterotrophic lifestyle, despite the fact that the carbon down there is old and kind of refractory compared to like surface ocean. And despite the fact that seawater is constantly moving through the crust, the microbial communities are distinct from it, and it really does vary across the aquifer. But just like we saw in vents, using both metagenomics and metatranscriptomics, we see that these communities are functionally redundant, and as I mentioned, they're quite motile. And one thing we also did, we did some really cool nanosims experiments. So again, adding labeled substrates like labeled inorganic carbon, organic carbon, nitrogen, and things like that. This is work I did with a former postdoc, Elizabeth Trembeth-Reichert, who's now a professor at Arizona State. And we used nanosims to measure single cell carbon uptake rates. And we found that the microbes just have this huge range of uptake rates and varied from those observed in deep marine sediments to those found at deep sea hydrothermal vents. And that really suggests a community that is ready to eat. They're ready to take advantage of a good thing when it comes by, and sort of far from just kind of sitting dormant. These microbes are ready to rock when they encounter a good thing. So I've told you a lot of science that offers insight into microbial life in the rocky subsea floor. It's probably more than you've wanted to know. I told you that they're phylogenetically and functionally diverse, that they thrive in these volcanic eruptions, that they fix a lot of carbon. They're cycling things like hydrogen and methane and sulfur and nitrogen more. And out in these ridge flanks, they're eating organic carbon. And I didn't really tell you, but they're also cycling things like sulfur, iron, and nitrogen. But I could also change the title of this slide, because I also want you to know that these things are also ecosystem services that these microbes are providing. And ecosystem services are benefits that we receive from the natural environment. And by identifying how natural processes support human well-being, we can begin to understand sort of this often unclear value that ecosystems provide us. And this direct link between natural systems and people is a really critical consideration for policy development across a number of issues, from the sharing of benefits to the responsible environmental stewardship. And I bring this up because while many of you probably work in spaces where this is obvious, right? Whether it's drinking water or biomedical work, it's not so obvious in the ocean sciences or those of us who are working in the natural world. And I think because many of us don't interact with this space, it's really important to be able to translate our basic scientific findings in that direction. So it can be awkward to talk about, right? So what I want to do is just kind of move these results into ecosystem services. So all of this, diversity and ability to adapt to an extreme environment, that's a really important ecosystem service in the sense this is a genetic resource for natural product and drug discovery, potential novel chemical compounds, industry, and other innovation. We can all agree, I think, that there's a huge amount of value in this particular service. What about these biogeochemistry ones, right? That's a little bit harder, I think, for people to connect with. But these are also important ecosystem services, right? They form new carbon in the ocean. They provide the base of the food chain that's important for nutrient cycling. If we think about how that all, for example, feeds into fisheries or sequesters carbon from the atmosphere. And I just think it's really important, regardless of what your science is, to try to think about your findings and making a little bit of a pivot like this. And as a deep sea oceanographer, it's really important for me to do. Most people will never interact with the environment I study, so it is not obvious how this realm provides ecosystem services to them. But the deep ocean provides over 95% of Earth's living space, and it hosts these incredibly diverse habitats and organisms that are essential to human and ocean health globally. And the reason I really want to turn my results that way is because increasingly the deep ocean, and especially the seafloor, are being looked at for these emergent human uses. And we need to be able to evaluate the impact of these activities with respect to these essential services and what it might cost us. So this includes things like deep sea mining, carbon sequestration, cable laying, seafloor warfare, and many more. And this is a real shift, right, to take sort of a fundamental exploration and discovery activity and move it into this more applied space. But I wanted to mention it in the hopes that you can see how important it is to learn how to translate your fundamental science of the seafloor to inform such activities. And I've been working with a group of scientists through a National Science Foundation network I lead to kind of focus in on the ecosystem services that microbes provide in the deep ocean. And the idea here is to really figure out what are the services microbes provide, what are the knowledge gaps in the deep ocean, what baseline data do we have, what are the best indicators for measuring them, and if we were to start doing particular industries or activities, how should we measure it, and what are some thresholds for harm? And so, for example, we have identified these 10 ecosystem services listed here on the left. We found that 4 of them have really great sufficient methodology for routinely measuring at sea in the moment, but only 1 or 2 have really good thresholds for knowing when an ecosystem is being harmed. This, of course, is a whole nother talk. There's a paper down there in the corner where from when we started this work, we're still working on the next iteration of this. But again, this is mostly just to highlight how this fundamental exploration and discovery research can be translated into this applied space. So now, just to close it out, let's take a breath. Let's thank the ocean. About somewhere around 30 to 40% of the oxygen you're breathing comes from the ocean, thanks to those phytoplankton. And we're going to make a pivot outward. So just in my final few minutes, I want to tell you one more story about how the deep sea is inspiring an entire generation of exploration here and well beyond Earth. And this is really a non-quantifiable piece. You'll never really be able to monetize this part of the ocean, but its mere existence of life in the deep ocean has inspired an entire new search for life beyond Earth. So we've just learned that life thrives in the deep, dark ocean of Earth. It is supported by microbial life. It's extracting chemical energy from Earth's crust. And we can't help be curious then to asking the question, what about on these other ocean worlds? Does life thrive beyond Earth? So just as our technology to help us study the ocean has improved, we've also learned more about oceans in our solar system. We know that Earth is no longer unique in having an ocean, and shown here is Earth with planetary bodies in our solar system thought to have a liquid ocean. Given we think water or liquid media is essential to life on Earth, we are following the water. And this discovery has just opened up a lot of possibilities for our search for life beyond. And most of these bodies are well outside. They're all in our outer solar system. They're mainly the moons of Jupiter and Saturn. It's very cold out there. These are icy covered moons. And this is one of them. This is Saturn's moon, Enceladus. It's over 900 million miles from home. It's about as wide as the state of Arizona. It's super tiny. But it's doing a very convenient thing for us, which is spewing its ocean out into space, which is what these little plumes are. And when the Cassini mission flew through the plumes of Enceladus, it had sensors on the spacecraft, and it detected some really interesting chemistry in that plume, including these 3 chemical species: hydrogen, CO2, and methane. And hopefully these compounds still look familiar. I introduced them at the beginning of my talk for a reason, because these, of course, are compounds that are important for microbes on Earth, for life here, and in fact, some really elegant modeling work from planetary science who continue to reanalyze the Cassini data suggests that Enceladus ocean likely hosts low temperature hydrothermal systems There are rocky particles in that plume, and there could potentially be sort of hydrothermal systems like what we host here on Earth. And of course, this raises, incredibly fascinating questions about what we may be able to find if we were to get a spacecraft that had life detecting or biosignature detecting technology on it. And I think it's just this really profound time in humanity when we can consider this possibility of detecting life beyond Earth. And it, of course, makes the imagination go wild. But I just want to close by reminding you that every second of every day right here on Earth, there are remarkable things happening in the deep ocean. This amazing video was captured while we were at sea in Costa Rican waters. A couple of years ago, we spent a lot of time watching these mama octopus slowly die. And we were very lucky to finally get to watch some eggs hatch and see these baby octopus make their way into the deep ocean. And this is probably something that's happening every day, every moment in this vast habitat we so rarely get to witness. And scenes like this will continue to inspire me, and I hope they continue to inspire you all to look a little deeper. And with that, I just want to thank the variety of agencies and individuals that have supported this work, and I'm happy to take questions.